Okay, so this is lab number 1 we're going to be working on, which is the blood lab. I just quickly want to review where you're going to find all your content. So over on the left-hand side of the course homepage, we have our syllabus, our lab schedule, my office hours, where we find all the lab guides, which we're going to use, per lab. So either print them out or store them to a thumb drive so you can fill out the appropriate answers. And then you have your study tool. We have under here, Zoom is where you will find the stored Zooms that I'm pre-recording. And then below that, we have lab number 1, lab number 2. So lab number 1 corresponds with the blood lab. And here is where I have uploaded notes in a PowerPoint form that has pictures of images I frequently use on quizzes and exams. And then also there we will find maybe some links to videos that are quite helpful. I found them to be very helpful. Under lab images, we'll go through some of those. Those are some images that we can also frequently use for quizzes and examinations. And then below that will be the quiz that will be due, by the end of the week. Generally, they're going to be on, I believe, Sunday nights. So that's where blood quiz will be located and where the cardiac quiz will be located. So first, what I'm going to do is change my share, or actually I can stay in here, I think, and go to the first lab. So I'm going to go to or actually, let me just stop my share and toggle back and forth. I think that's going to be easier. I can go to, hold on, there. There, I think it's easier if I change my share. Okay, so I'm going to go up to the top here, and this is where we have our first lab. So this includes all the exercises and tells you whether you to just identify or know functions or something about this particular topic or exercise. And the first one is blood plasma. So here's to determine the function of the following components of plasma: water, albumin, fibrinogen, gamma globulins, the percent, of blood plasma found in blood. What is blood? It's 55%. So what would be the percentage of plasma of blood? And then below, when we're down here, when we're looking at the formed elements, it's going to ask you, well, what percentage is formed elements of blood? And that would be 45%. And also at this point, we're going to include your pH range. So this is the exercises that we perform. And then we're going to apply that information to the images, and using the notes. So let me switch back. I'm going to toggle back to the notes. Hold on just a moment. And I'm not always going to do this, but at the beginning, I frequently do this so that we can kind of go through together and see where this information is coming from. All right, so it's just preparing, it's uploading the information. So let me just wait for that a moment. So there's a lot of information in the PowerPoints that I provided you. I think that, it's depending on the instructor you have and who you have for lecture. This really helps bring you up to speed of what you need to know for lab, because lab and lecture generally will reinforce each other, but sometimes the schedule of topics is not going to exactly coincide. So the answers to all those, most of the fill in the blanks, I would say, in those lab exercises that I just showed you, will actually come from the blood. So I'm going to go through these notes and then going to let you apply these answers and use these to put into the gaps. And I'll give you some examples of how we're going to fill in those lab exercises given the notes. So here I have the PowerPoint notes and talks about blood. So blood functions, transports vital substances, maintains stability of interstitial fluid, distributes heat. And that's a big function of water, the water component of blood. It's composed of formed elements, 45%. Formed elements are red blood cells, white blood cells, and platelets. And plasma is 55%. Right? The adult blood volume is about 5 liters, give or take. Of our formed elements, we have red blood cells, white blood cells, platelets, erythrocytes, leukocytes, and thrombocytes. So plasma is composed of primarily water. 90% is water. We transport a lot of electrolytes. Remember, electrolytes are ions in solution, and ions are elements that gained or lost electrons. We have proteins that help maintain osmotic gradients, especially albumin. We're going to identify 2 other proteins in plasma in the lab exercises. And then we have things like nutrients, such as glucose, amino acids, lipids, really anything that has to be transported from one organ to another, is going to be put into blood to be transported from one place to another. Transports gases, actually gas is just dissolved in the plasma, like carbon dioxide and oxygen, and it transports different waste products like urea and uric acid. So blood plasma, the water acts as a solvent, absorbs heat, helps distribute heat very well, actually. Albumin maintains an osmotic pressure or gradient. Fibrinogen acts in clotting and helps form fibrin threads, but you can just say acts in clotting, that's fine. And gamma globulins are antibodies. We'll look at the 5 different classes of antibodies later within the course. All right, so characteristics of red blood cells. Not that you need to know everything about this, but this helps you formulate a good picture in your head as to what you're looking at here, how you might identify them on a slide histologically, and then functionally. So blood cells originate in red bone marrow. Remember in AMP1, we looked at spongy bone, and we said the 2 major components of spongy bone are trabeculae and red bone marrow. Well, red bone marrow produces all blood cells, not just red blood cells, but white blood cells and platelets and so forth. They are biconcave discs, so they're kind of hollow in the center. I call it the lifesaver appearance because they look like those little candy lifesavers. And in the center, we have hemoglobin. And hemoglobin, is a quaternary protein structure. It has what's called a hemigroup in the center. And in the center, that is an iron molecule. And to the iron is what oxygen will bind to, or oxygen binds to the iron molecule. And therefore, red blood cells are capable of transporting oxygen as well as carbon dioxide. Given that concave shape to it, right, because it has a nucleus when it passes through its different developmental maturation stages, but at the end, it releases its nucleus, and red blood cells are actually anuclear, and therefore they have a limited lifespan of about 100 to 120 days. That concave shape gives it flexibility, so it can pass through small blood vessels, like capillaries, and bend a little bit and squeeze through and pop back open again. The red blood cell count, the number of red blood cells per microliter of blood, has a direct correlation to the amount of oxygen that you're carrying. So the number and quality of red blood cells is a direct indication of the blood's oxygen carrying capacity. Functionally, it transports oxygen and carbon dioxide normally, most of the time. It can bind to other molecules like carbon monoxide, but that would be kind of abnormally. Typical range is about 5 million, per microliter, in males, females, and children. So around 5. Right here, we're looking at what would be what's called a peripheral smear. You're looking at some blood cells that are stained and prepared on a slide. And you can see how they can be actually translucent in the center. They're the most abundant cell you will see. All right, so terms you should be aware of. Anemia is a lower than normal number of red blood cells. Anemia is a very broad category. And we touch back on this later, some of these like broad categories of things like jaundice, well, jaundice and just yellowing of the skin due to higher levels of worn out red blood cells, degradation portions of it, is a very broad category. And so is anemia. right? But in general, it means that it's a lower than normal number of either normal red cells or, they're abnormally shaped. Etiologies will vary. At one of the last pages on here, I identified different types of anemias and what they're often due to. So polycythemia, is the opposite. That's a higher than normal number of red blood cells in circulation. Sickle cell anemia is an unusual type of anemia, and it's discussed at the end of this also. Sickle cell anemia occurs when you have a substitution of one particular amino acid at the 6th position for a different one. And that one particular substitution in the amino acid chain changes the way the cell makes its hemoglobin, this protein. And the effect that it has multiple effects, but one of them is when you're under low oxygen content or tensions, where you either have low oxygen availability, like at high altitude, the cell actually twists and turns and takes or assumes kind of a boomerang kind of a shape, or a sickle. And that's what we're looking at here on this little slide. And when that happens, you change the ability, you change the surface area, so therefore you change oxygen carrying capacity, binding to hemoglobin, as well as it changes the plasma membrane of these red blood cells, and it makes them more fragile, or changes the fragility, and it makes them stickier and things also. So you not only become stickier and more likely to maybe block off little capillaries around joints like the elbows and the knees that can be quite painful. But you also limit the amount of oxygen carrying capacity, so you become a little hypoxic or terribly hypoxic, depending on the degree of sickling that you have. There are full-blown sickle cell disorders, and then you can acquire a carrier condition, which means that some of your cells sickle, but they don't generally sickle to a high degree, like if you had what's called full-blown sickle cell anemia. We look at that in one of the last labs of the semester when we talk about the ways in which we inherit different disorders, and what it is to be normal, what it is to be kind of a carrier, of sickle cell anemia, and then to be full-blown sickle cell. All right, so then white blood cells. White blood cells are called leukocytes. Right? They protect against disease in different ways, and each cell kind of has its own specific function, and therefore can elevate or depress under different conditions. So we have 5 different types. We have our granulocytes and our agranulocytes. The granulocytes, your nice little mnemonic is BEN, basophil, eosinophil, neutrophil, and the agranulocytes are lymphocytes and monocytes. Right. So the first one, we're going to look at our neutrophils, also called polymorphonuclear cells. Right. And here we have an example of a neutrophil. White blood cells generally are larger than red blood cells, generally. And their nuclei take up more intense staining capacity. Most of them will take either a dark purple or moderate purple. And then the granules, if there are any, usually will take up a purple stain. So looking at this cell, you'll see that it kind of has what's called the sausage link appearance of a nucleus. They're in segments and held together by these little pieces of nuclear material that holds them together. So think of like sausage links, all strung together. And that's what we call polymorphonuclear. It has abundant cytoplasm. They're kind of in a light, in this staining, a light lavender, kind of a light pink. And you have granules that you can see, right? They generally are taking up kind of a darker purple stain, but you can still see a lot of cytoplasm in the nucleus and the segments of the nucleus here. So those are things to keep in mind to help you identify a neutrophil on a slide. So its main function is phagocytosis. They are, called nets. They trap and kill microorganisms and inhibit spread throughout the tissues. All right, they are capable of phagocytosis, which is an active process of engulfing some kind of bacteria, and that's generally what they're going to engulf. Right, they are the first to arrive and to elevate when you have a bacterial infection. They can generally phagocytize one bacteria before they succumb to it. One of their lysosomes will merge with that phagosome, which it just phagocytized, created a vesicle around, and the two vesicles merge, and the lysosomes secrete the digestive enzymes onto that bacteria and help kill it. Other names of neutrophils are segs and bands. If you already work in a healthcare environment and you look at some of the results of hematology reports that come back, you'll see that they'll give you a neutrophil count. They do it by percentage and by actual number, and they'll often give you a percentage of the segs versus the bands. The segs means the nucleus is segmented like this, so that's the percentage of mature neutrophils that you see. And bands are young ones, that are not quite mature yet. And the nucleus is in the form of kind of like a bent hot dog, in the shape of a hot dog. And it's the maturation stage just before it starts twisting off and produces these little segments. So it's the first to arrive at infections and the most abundant. Generally about 50, I would say a nice rule of thumb would be about 55 to 65% of all white blood cells would be about neutrophils. Right? So they're elevated in bacterial infections. They're the first to arrive. They're the most abundant normally. Right, then we move on to eosinophils, so EO orange, right? Eosinophils have a segmented nucleus, but it takes up kind of a different type of a stain because we use a different kind of stain to really see the eosinophils, or to try to visualize them and count them. Their granules will often take up kind of an orangey, pinky kind of a tone, and it's the only cell to do so. So if you see a white blood cell that has granules that are rather pale staining and abundant and kind of pinky orangey, especially on the models, we'll see that they're orangey, that's an eosinophil. They're elevated in allergic reactions, allergies. They help defend against parasites. And really the only one that will elevate in the presence of parasites. It's kind of a good board question. Generally, only about 1 to 3% of all the white blood cells or leukocytes will be eosinophils. So they're elevated in parasitic worm infections and allergic reactions. All right, then we go on to basophils. Now, basophils are very infrequently occurring. It's normal to have maybe none to maybe 1 out of 100. So 0 to 1% may be a basophil. Right? And they're very easy to confuse with a lymphocyte, but I'm going to tell you what makes it differentiates them. And with a good microscopes, you can see that. Generally, if we're going to ask you to identify basophil, we're going to use a model, which is what I call cells on stick models. But occasionally you'll see a slide, and if you see a cell like this, which is A, then and you very, very difficult to see a nucleus. All you see is a lot of purple. But upon really looking at that, you go, those are actually purple little granules in there, right? That's a basophil. Right. A lymphocyte will have a solid dark purple nucleus, but often has a halo of clear kind of light lavender cytoplasm, either off to one side, or completely around the nucleus. So sometimes it looks kind of like a crescent moon kind of a cytoplasm, or it will be all the way around it. But you will see a solid nucleus, not with granules. With a basophil, it's the abundance of dark staining granules really obscures your ability to see a nucleus. And that's one of the ding, 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 tip-offs that you're looking at a basophil. They're active in allergies and inflammatory conditions. They secrete histamines and heparin. Histamines increase blood vessel dilation and leakiness, and heparin inhibits clotting. And so it's normally you have maybe 0 to 1% of 100 white blood cells that you look at will be a basophil. And that's what a hematocrit is you start getting, when you do an analysis of blood, you get counts of your red blood cells and your white blood cells. And of your white blood cells or your leukocytes, it's going to break out the percentage of your 5 classes of white blood cells. And so normally you might have just 0 to 1 of a basophil. All right, then we have monocytes. So you gave me mono because frequently, if you look at if the cell turns in such a way where you can see the nucleus, it often takes a U shape. And I call them the big mothers because they're huge cells compared to the other white blood cells. You can almost take about 4, if not more, red blood cells, and put them inside that monocyte. That's how big it is. The nucleus, is moderately purple staining, and the cytoplasm, is about 40 to 50% of the volume of the whole cell. It's very large. It's not abundant in granules. It's rather light lavender. It's big. It's a monocyte. All right, so it's the largest of all the white cells. They arrive at an infection site after the neutrophils. So the neutrophils will elevate first and rapidly when the bacterial infection. Monocytes will follow up a few days later, and they'll start to elevate above their normal percentage rates. They leave the bloodstream, become macrophages and dendritic cells, in the skin, if we find them there. And they can act as what's called an antigen presenting cell. And we'll talk about that when we get to the immune system. When a monocyte phagocytizes bacteria, but it can phagocytize all kinds of things. It can phagocytize very small parasites if need be. It will phagocytize viruses. It helps break down dead cells, like it's very active in breaking down and recycling erythrocytes, and helps kind of phagocytize areas that are healing when you've had, let's say, broken bones or repairing tissue in the body. In any event, they can be an antigen presenting cell because if they phagocytize a virus, it can take the antigen from the surface of that virus and put it kind of on the surface of its white blood cell and take it to the lymphocytes and be able to say, hey, this is the antigen we want to create antibodies toward. And that's how your immune system works. Normally, they're about 3 to 9%. I'd say the average is about 5%. Then we have lymphocytes. Right? We have 2 types of lymphocytes. We have T cells and B cells. Of the T cells, we have what's called T4s and T8s, and then we have the B cells that become plasma cells. And then we also have another class called like natural killer cells. And they're very active when it comes to cancer cells and being able to phagocytize or actually combat them. They don't really phagocytize the same way, but they act differently with cancer cells. We'll talk about that when we get to the immune system. So lymphocytes, here we have a lymphocyte, a mature lymphocyte. The nucleus is about the size of a mature red blood cell or erythrocyte. So that's about the same size. It has cytoplasm surrounding it. And I'll say that that's rather dark staining. Most of the cytoplasm we'll see around a lymphocyte will be a little paler than that. But it looks very different than a basophil, where you can't really distinguish the nucleus and there are granules all over. So a mature lymphocyte nucleus should be about the same size as a red blood cell. Large nuclei take up almost the entire volume of the cell, about 90 to 85%, and often has a light violet halo of cytoplasm surrounding it. You have T cells and B cells. We'll look at those and distinguish between those and natural killer cells when we get to immunology. But right now, you should just know that we have T lymphocytes and B lymphocytes, or T cells and B cells, both important in immunity. They just function differently. But it is the B cells that produce the antibodies. And normally about 25 to about 33, 35% of white blood cells will be lymphocytes. Here, we're just going to elaborate a little bit. With the lymphocytes, T lymphocytes, we have T4 helper cells that release the cytokines. The T8 cells, lyse virally infected cells and tumor cells and transplanted tissues. Natural killer cells kill tumor cells. Virally infected cells, I should say. And some bacteria, fungi, and parasites, but mostly they're very, very active with cancer cells or tumor cells. I think it's thought that they probably are active every 6 months, that a natural killer cell is probably eradicating some kind of cancer cell in your body. And then the B lymphocytes become what's called plasma cells. to form antibodies or make antibodies. This isn't in red. Most things in red are really what we're going to highlight. Just know right now that lymphocytes, we have T lymphocytes and B lymphocytes, maybe some natural killer cells that would be helpful, I guess. But when it comes to them specifically functioning, just know right now maybe that the B lymphocyte is the one that produces the antibodies. Because when we go back and we look at some of the, the beginning of this exercise and filling in these lab exercises, it mentions the gamma globulins, and those are part of antibodies. Okay, this is just a picture, helps kind of compare and contrast leukemia versus normal blood. Leukemia is generally, there are different types of leukemia, but usually it's a proliferation of the lymphocytes. So here we have, let's say, 1/4 of the field that we see on the right, where we have the ABCD. Right? You can see lots of red blood cells there. We can see some white cells. We have an A, the B, two Bs because they're both neutrophils. Right? A is a lymphocyte. C is pointing to some platelets. They just like little purple dots there. That's what the platelets are. Right? And if I drew that into quarters, 1/4 of that big field, on that whole slide on the right, that's what the left-hand side is. So over on the right side, we're looking at 4 times as much as we're looking at the left. And out of all those 4, we really only see 1, maybe 2 lymphocytes might normally be found there. If we took only 1/4 of that field and magnified it, in leukemias, which are generally, proliferation of lymphocytes, look how many lymphocytes we would see. I mean, they're hugely abundant, and that's just 1/4 of what's on the right. So leukemia, once you have some of these abnormal conditions developing, unless it's very, very, very early stages where you can't really see too many, an increase in the lymphocytes, it might be overlooked. But once it starts getting fairly developed, you have so many more lymphocytes than you normally should. It's generally fairly easy to at least diagnose as some kind of abnormality, and then you have to start specifically looking at the different types of leukemias, myelogenic or lymphomic, the different types of leukemias. So functions of white blood cells, they protect against infection. They are capable of what's called diapedesis. Here we have a little capillary, and white blood cells can squeeze through the squamous cells, where the squamous cells abut with one another, in a capillary. We'll look at that a little bit more when we get to the blood vessels, which is 2 labs away. So that's called diapedesis. I go through these kind of quickly. This is the first one so that you understand where a lot of this content is. And then, I will move on to covering the lab exercises, but I want you to be familiar with these notes and how, if you don't participate in the Zoom or you don't want to watch the Zoom, you can just look at the notes and fill in most of your answers, like 99% of them. A procedure used to count the number of white blood cells per cubic milliliter, is a hematocrit with your CBC, which is called the complete blood count, with a differential. That means it's going to take 100 cells, white blood cells, and it's going to categorize them, into their neutrophils and lymphocytes and monocytes and so forth. Normally, you have 5,000 to 10,000 white blood cells per cubic milliliter of blood. So the 5,000 to 10,000 range per cubic milliliter. When you have less than 5,000, that's called leukopenia. So enia, means lower than normal. So leuko, white blood cell, low white blood cell count. Leukocytosis is a higher count. So if the count is above 10,000, we call it leukocytosis. And that's not uncommon when you have acute infections. I mean, sometimes they'll just peak like that temporarily. And then if you run the blood work, about 2 weeks later, a lot of times they'll start to diminish. So it's not uncommon if you have an acute infection, vigorous exercise. If you're very dehydrated, then the number of cells in a cubic liter, a milliliter, or a cubic milliliter of blood will kind of be exaggerated because you're so dehydrated. You rehydrate and run the blood test again, it's normally back into normal ranges. The differential WBC lists percentages of the different types of leukocytes. Neutropenia is specifically a lower than normal number of neutrophils. Count less than 4,000, can put you at very high risk of bacterial infections. Remember that neutrophils are the first to combat bacteria, and they're generally the most abundant white blood cell. And if you don't have very many of them, then you're relying more on your monocytes, and it takes them a while to become elevated. So neutropenia, you are at more risk or higher risk of profound bacterial infections. All right, so when this is a good picture to keep in mind, to use this in lecture also. Out of blood on the top, okay, we have 45% formed elements, 55% plasma. So we start with that major division. Right. And then let's look at plasma. Okay, out of that 55% plasma, we have electrolytes, water, which is 90 to 92%, proteins, waste products that we transport, nutrients, vitamins, hormones, gases that we can transport. Out of the proteins, we have albumins, globulins, and fibrinogen. Albumins are like albumin, which we looked at in the chemistry portion of AMP1. We said maintains an osmotic gradient in plasma inside a red blood cell. It helps to balance pH, but we'll talk about that later. Globulins are active in antibodies, and fibrinogen in clotting. And then here at the 45% formed elements, we have platelets, red blood cells, about almost 100% is going to be red blood cells. Technically, it's about 95%. Then we have white blood cells. And out of the white blood cells, we have our neutrophils, eosinophils, basophils, monocytes, and lymphocytes. You don't have to know the exact percentages, because those percentages will vary per kind of publisher of a textbook, but also given each lab in each facility, each hospital. The normal ranges are generally dictated by the lab director, who is usually an MD, and it will vary sometimes regionally, but they're basically about the same. What I would know is what is the most abundant and what's the most least abundant. And then you need to know, well, what do they respond to specifically? Is it more, parasites? Is it first to elevate in bacteria, second to elevate in bacteria? Monocytes, they phagocytize really anything. They're garbage collectors. Lymphocytes, are the T cells and the B cells. They are active in what's called specific immunity. Eosinophils respond to parasites and produce antihistamines. Basophils produce histamine and heparin. So be able to kind of compare and contrast them functionally. Blood platelets. Platelets are also known as thrombocytes. They're actually cell fragments. They kind of exfoliate off the surface of what's called the megakaryocyte. And that's why you have all those little dark purple fragments all over. Right, so they are active in clotting. Clotting helps repair damaged vessels, by sticking to the surfaces and bringing them together. If it's, it depends on whether it's an extrinsic pathway, that's what it is. An intrinsic pathway, has clotting, but for different reasons. The fate of blood clots. A thrombus is an abnormal blood clot that's stationary. An embolism is a clot of some kind that breaks free and is mobile. Resistance and opposition to blood flow. So something we often talk about are different factors that affect the thickness and how well blood flows. We talk about this again when we get to the blood vessels, because we talk a lot about blood pressure, and things like that. But opposition to blood flow, like water passing through a hose, is resistance. So what can affect the opposition to blood flow, maybe, inhibit it or resist it somehow? It depends on a couple different factors. The diameter of the blood vessel, the more narrow a blood vessel is, the thinner the little passageway is, the more resistance it imparts. And the larger diameter, like a great big hose or a pipe, produces less resistance. The length of the blood vessel, the shorter the vessel, the less opportunity for resistance. Or think of it this way: friction of blood, passing, across the interior of a blood vessel wall. The shorter it is, the less opportunity for resistance. The longer it is, the more opportunity for resistance. And then the viscosity, which is the thickness and the stickiness of blood. how much water, is there in the blood plasma? So when we talked about that 55% plasma being 90% water, hydration levels have a big impact on the thickness of your blood. So the thicker it is, the more viscous it is. So blood water levels, will have a big impact on the viscosity. How many red blood cells there are, am I polycythemic? Do I have more than a normal number of red blood cells? Which means that ratio of formed elements is no longer 45% in plasma, 55%. It might be 50-50 or even 55% formed elements and only 45% plasma. So the watery portion starts to be offset by the number of red blood cells. And what is the ratio of, so therefore, what is the ratio of plasma to the formed elements? Is it the 45-55% normal ratio, or is that being thrown off? So here we increase viscosity. If we increase viscosity, we decrease blood flow. That's what BF is, blood flow. If we increase the number of red blood cells, we decrease blood flow. Therefore, we increase our viscosity. If we decrease plasma, right, then we also decrease blood flow and increase viscosity. If we increase our RBCs, we initially, at least at the beginning, as we start increasing our red blood cells, like in polycythemia, we initially start to deliver more oxygen. But after a while, when the number of red blood cells becomes extraordinary, now it's the blood's become too thick. I mean, it's one thing to increase the number of red blood cells, as people do in blood doping situations. Some athletes, you start to increase the number of red blood cells. And hey, if you increase it by 1 to 2%, you are really circulating and transporting more oxygen effectively. And you're not really changing the ratio of formed elements to plasma all that much. But if that red blood cell number starts to get higher and higher and keep getting higher, and now you have more formed elements than you do plasma, you're not delivering, the blood flow is flowing much more slowly, and you're not delivering as much oxygen. So ultimately, you are delivering less oxygen over time. Even though you have more red blood cells, you're just not flowing as fast. So here we have erythropoiesis is the formation of blood cells. Right. The stimulus is a negative feedback loop. We'll look in the exercises. The stimulus is hypoxia or anemia, but anemia really is hypoxia. It's a lower than normal amount of oxygen being transported, really. That's what hypoxia really refers to. Anemia is just a decreased number of red blood cells. Production side are the kidneys. The kidneys release what's called erythropoietin, or EPO. That is the hormone. The hormone travels and targets the red bone marrow. And the effect of the red bone marrow is to increase red blood cell production, so to produce more red blood cells and therefore increase your oxygen carrying capacity. This is a negative feedback loop. What we start with, our stimulus, is low oxygen or low oxygen carrying capacity. We end with higher or normal oxygen levels. Okay, then when it comes to blood groups, this is how we ABO blood type. I did put a video up there for you to watch, and I think that's a pretty good one. It's this young, I think it's the same. Let me just make sure it's the one. I'm pretty sure it's the one I really like. It's this kind of younger guy, and because some of those videos out there, you get these old dudes, and well, anyway, I'm old, but you just don't relate to it quite the same way. And this guy is a little bit younger, and he talks about, and he shows you some examples along the way of how you ABO blood type, what it means, and then how you match blood transfusions to it. Not something you need to know now. We generally reserve our questions for, what is the blood type? What are the antibodies you would make? Maybe who could you donate blood to? But I'm not even sure if there's any specific questions like that in the exercises. But what I would do is, if you're going into nursing, is save that, because that I found helped some of the nursing students that I stay in touch with. And, they were asking me to have a review for ABO blood typing. And we were kind of doing that like Zooming-ish, for this past year or so. And then I found this video, and it was really good. And they just watched that a few times, and they were able to kind of quickly match up, because in nursing, you have to know who can you donate to and who you can receive blood from. And he puts it in this chart, that is good for later. Not is TMI right now, but it would be good for use later. Okay, so antigens and antibodies, we have to know what this stuff means. Well, agglutination is an antigen-antibody reaction. It's when you see the blood actually kind of curdling and clumping, and it means that antibodies have found a corresponding antigen to bind to. Right? So an antigen is a chemical compound found on a cell surface. It is unique to these cells and to different classes of cells. Viruses have certain types of antigens, and bacteria too, where it has this chemical marker essentially on its cell surface. And that's how your body cells know, is this one of my self cells? Is this one that's supposed to be here? Or is this a foreign invader? Because they don't have eyes, they can't see anything. All they can do is respond chemically, right? So an antigen is just this, it's kind of like, a little marker, I guess that's the best way to describe it, on the surface of a cell that lets your body cells know whether they're normal cells that should be in your body or not, whether they're foreign cells. An antibody is a protein that attaches to a specific antigen. So an antibody, is specific to bind to a particular antigen. It's like you have a lock and a key, and you only have one key that can generally fit into a lock, and that's it. If you want to change the locks, then you got to change the key. Right? So they are unique and specific for one another. So when you make antibodies, they are specific to fit and interact with one antigen only. So when it comes to blood typing, we have what's called the International ABO blood typing. You're either A, B, AB, or O. And then our other designation is whether you are Rh positive or Rh negative. Now, we're testing here in this case for the presence of A antigens, B antigens, or what's called the D antigen, or the Rh. The A and the Bs and the Rh are of a specific shape and size. And that's where we start by classifying blood cells. They are the fastest and the easiest to have mismatched blood, and to have traumatic hemolysis taking place with mismatched blood. So it's important that you understand and carefully identify a patient's blood type, and match it to blood that can be donated to them. Now, these A and the B and the Ds, or the Rh antigen, are only 3 of about 400 different antigens that can be on a cell surface. So you start by just identifying the presence or lack thereof of these. And then there's other forms of analysis to find out exactly what antigens are on your red blood cells or the patient's red blood cells, and what antibodies have they already started making. All right, so to understand all this, you have to understand why we call something A blood, B blood, AB blood, or O blood. So if you have A antigens on the cell surface of a red blood cell, so here in the upper left-hand corner, we have a red blood cell, and you see you have these little orange inverted little indented, blocks on the surface of the red blood cell. And like I say, we're going to use that as an example of what an A antigen looks like. Right, that's the shape. So if we have A antigen on a red blood cell surface, we call that blood type A. And when we have A antigens on our cell surfaces, we will not automatically make any antibodies to those, but we will make antibodies to the opposite, and that will be B antibodies. So if I am blood type A, I have A antigens on my cell surface, I make anti-B antibodies, or B antibodies. And you see how the tips, that happens to be an IgM, antibody, but you see how on the tips you have green little binding sites there. And you see how that green can't possibly interlock and bind with those oranges. They just kind of bounce off one another. So B antibodies cannot bind to A antigens. All right, then over to the right top, we have type B blood. And look, this is type B blood because we have B antigens on our cell surface. And those are those green little cubes that are on the surface of that red blood cell. So when those are present, B antigens on the cell surface, we call it B type blood. And when if we're blood type B, we make A antibodies. So look at those antibodies. Now they have little orange tips on them. And there's no way that that little orange little arrow tip is going to bind with the green arrow. They're just going to kind of bounce off one another. All right, so we have no worry that the antibodies that I'm going to automatically make at about 4 months of age from our liver, right, are going to bind with the antigens on our own red blood cells. Right? All right, then we're going to go down to the bottom, type O blood. If you have neither A nor B antigen on your cell surface, I call it being like a bald red blood cell, that makes you type A blood or type O blood, or that is what determines type O blood. So you have neither A nor B antigen on your cell surface, and therefore you make both A antibodies and B antibodies. You have no risk of those antibodies binding to any of your own red blood cells because you have neither A nor B antigen on your cell surface. And then lastly, we have type AB blood. And so guess what that means? That means that we have both A and B antigens on our cell surface. Look at that. We have A's and B's on the cell surface. That's how we know, or that's why we will call it type AB blood. And when you're A and B blood type, you make neither A nor B antibodies. Because if you did, then those antibodies could bind to the antigens on your own red blood cell surface, and you could end up killing your own red blood cells. And you don't want to do that. So normally, forever and ever going forward, you don't normally make antibodies to any antigens found on your body cells, period. Otherwise, you'd be killing off your own cells. And that can happen because that's, what different autoimmune disorders, are about when you, for many different types of reasons, is you start making at a lesser or greater degree antibodies to some of your own body cells. But we'll talk about that in immunology. Okay, so to be blood type A, you have A antigens on your cell surface, you make B antibodies. If you're blood type B, you have B antigens on your cell surface, you'll automatically make A antibodies. If you're blood type O, you have neither A nor B antigen on the cell surface, you make both anti-A and anti-B, or A antibodies and B antibodies. And if you're blood type AB, you make neither A nor B antibody. Now, this is where this other video is pretty good because he shows you, he takes some of these A antibodies and B antibodies and he throws them into the plasma surrounding AB blood. And then you can see how the A antibody can bind to an A antigen, or a B antibody can bind to a B antigen. And when that happens, it gets all clotted and glumpy looking, and that's what agglutination is, when you have an antibody antigen interaction. And that's why you don't want it to happen. You want to match up blood correctly because you don't want to clot up all your blood and have them start to have these agglutination of your red blood cells and start killing them off. Essentially they'll hit your spleen, and then you spike a fever and have bad back pain and become jaundiced and could die. Okay, so, and this is the premise of how you identify a blood type, because when you test for blood types, we don't know, you have an unknown blood type. Most of us know what blood type we are. I know I'm an A positive, right? But if you don't know what someone's blood type is, you have to go through a series of steps to determine which antigens are on the cell surface. That's got to make sense. If I have a blood type A person, right, but you don't know it's blood type A, you just, you don't know, right? So this is what you can do. You can take some A antibodies, because you can have them manufactured. You put a little droplet, and you get a little eyedropper, and you put a couple drops of what is our A antibodies in solution, and you put it into a blood sample. Right? And if those A antibodies are binding to an A antigen, if the A antigen is there, the A antibody is going to bind to it, and it's going to start that clumping process. And that's what's called agglutination. So if I put A antibodies into an unknown blood sample and I start seeing clumping, at that point, I at least know I have an A antigen on the cell surface. That much it tells me. All right. Then you get another sample and you take some B antibodies in solution and you put it in that blood and you look for the same thing. Does it, you have to mix it up, give it a minute. Does it clump? Oh, it clumps too. Then I must have B antigens on that red blood cell surface also, because B antibodies only bind to B antigens. So at that point, you know I have A antigens and B antigens. I must be blood type AB. You know, if I put the B antibodies in there and nothing happens, it just kind of looks like melted cherry jello, you know, and it might be streaky a little bit, but it's not like clumpy or granular looking. Then it means you don't have any B antibodies. You must be blood type A. You would do the same by administering, you know, either Rh sera or D antibodies, Rh antibodies, and you put it in a blood sample and you look to see if you had the D or Rh antigen on the cell surface. If you have that antigen and you get clumping, it means you're Rh positive. So you would be A positive or B positive or AB positive or O positive. If there is no clumping, it means you don't have that D antigen or Rh antigen on the cell surface. It means you're negative. You're negative for the presence of the D antigen or Rh antigen. Right, so Rh positive and negative is either you have this or you don't, the Rh or the D antigen. Okay, so here they're just showing you how here if you have B antibodies, you know, if we introduce now, you know, like an A antibody into the solution, you know, because we have a blood type A, we have, you know, A antigen on the cell surface, you have B antibodies. If we administer some A antibodies and then we mix them together, that takes us up to B up there. And you can see how the A antibodies will bind to the A antigen because they have complementary interlocking effects. And so something, I mean, the picture in D is highly magnified, but that's essentially what's happening during agglutination. Right, so the ABO blood group is based on the presence or absence of two major antigens. As I described before, you can kind of read through this on your own. If you're blood type A, you have the A antigen, you make the antibodies, so forth and so on. I just described that. Right, and so therefore, if you are blood, you know, if you have blood type of your recipient, you're trying to match donor blood to a recipient, right? When you donate blood or when you transfuse blood, something to keep in mind, you only transfuse the red blood cells. You spin it down and you separate the erythrocytes from everything else, and then you just put it in saline. So you don't have to worry about if you donated blood, someone's donated blood, you're not going to transfuse what's called whole blood. You're just going to transfuse packed cells, which is just the red blood cells. So I don't have to worry about what antibodies this person made or what they have in there. All I'm getting are their red blood cells. All right. So if I, you know, if the recipient is type A, right, and they make B antibodies, right, you can either receive blood from a type A or an O person. Why? Because A has A antigens only on the surface, right? So just like you do, and type O has neither A nor B, right? So you, even if a blood type A person is making B antibodies, there's nothing to bind to on the red blood cell surface of a blood type O. And the same for B. B can acquire from B and O. If you're AB, you know, then your preferred donor is another AB. But you know, you can, in circumstances, transfuse an A or a B or an O. And if you're blood type O, you can only receive from O, because remember, blood type O makes A and B antibodies as the patient, as the recipient. So it can't have any red blood cell introduced to it that would have an A or B antigen on the cell surface. So O to O. Rh blood group was named for the rhesus monkey. Rh positive means the presence of the protein or other Rh antigen on your red blood cell surface. It's called the D antigen, but you can just call it an Rh. You know, if you're Rh positive, it means you have it, which means you're not going to make any, Rh antibodies if you have the Rh antigen on your cell surface. If you're Rh negative, though, it means you don't have that antigen. And it's not one of those that you automatically make. The only antibodies you automatically make are anti-A's and anti-B's. Every other antibody, you have to be exposed to something first. I mean, that's the whole point of vaccinations right now, is that I don't make antibodies to a virus unless I get exposed to it somehow, either live or dead, or we're going to use these other newer methods like, messenger RNA type things where we can teach the cells how to find it. But in any event, if I'm Rh negative, then if I get exposed to Rh positive blood somehow, then I may start making antibodies to the Rh. So I may eventually over time form Rh antibodies or D antibodies if I get exposed to Rh positive blood if I'm an Rh negative person. The seriousness of the Rh group is evident in the fetus that develops the condition erythropoiesis fetalis, or hemolytic disease of the newborn. And you can read through this. I think it's rather interesting because this is why you need to know whether expectant mothers, if you're pregnant, whether you are, the mother is Rh positive or Rh negative, very, very, very early to help protect, the fetus. And what happens is if you're Rh negative, the mom is Rh negative, and the fetus is Rh positive because dad is maybe Rh positive, then mom can start to make antibodies once exposed to maybe some of the Rh positive blood of the developing fetus. And this can happen. It's usually not at risk for the first pregnancy because, it takes a little while, and often you don't get mixing of blood like that until birth, maybe upon amniocentesis, trauma, actually vaginal birth, where you have some mixing of the blood. But once the mom is sensitized and she starts making those antibodies, there's no coming back. Right? So you will, if you have a subsequent pregnancy, then those antibodies on the second child, if that child is Rh positive, can cross the placenta and start attacking the red blood cells, of the fetus, during development. Embryological development, so during pregnancy. And that's what erythropoiesis fetalis is, or hemolytic disease of the newborn. If you know that the mom is Rh negative, doesn't matter whether that fetus you can confirm is Rh positive or Rh negative, you can prophylactically start them on kind of IgG, immunotherapy to bind up and keep mom from ever being exposed or detecting the presence of an Rh positive blood from a fetus, and keep her from ever making those antibodies. If she does, and she starts making those antibodies, I mean, there are some things you can do. There are different types of transfusions. You can do umbilical transfusions. You can take a mom's blood and transfuse it and try to interact with the Rh antibodies that she's making. It's kind of a long discussion, but anyway, there are some things you can do, but it's better to know and never produce the antibodies to begin with. There's like a Rose test and things like that to help you identify if she does have any antibodies, how many. So this is an FYI down here, and it talks about erythropoiesis fetalis. You can read that on your own, blah, blah, blah. And to one of the last of the two PowerPoints here is, this is essentially what you're looking at when you're doing the unknown typing of blood. So here in well number 1 over, or let's say to the left-hand side where you have anti-A, anti-serum is administered to a blood sample, and you stir it and you watch for it. You see how you have all that clumping kind of going on? And that's fairly obvious. Sometimes it's not quite so obvious, but that's obvious. Right? That would be a positive reaction. So if you put in there A antibodies and you get clumping, then there must be A antigens on those red blood cell surface. So that is a positive reaction. Okay, so far I know there's an A antigen on the cell surface. Right. Then we put B antibodies in there, and you stir it and you stir it, and you see how it kind of looks like melted jello? That's a negative reaction. So that means there are no B antigens on the cell surface. Then, but we're not done. It's another step. Then we put anti-D or Rh antibodies in there to determine if you're Rh positive or Rh negative. And look, we have clumping again, which means that the D antigen is there, or the Rh antigen is there. That's a positive reaction. All right, so we're A positive. And here is the control group over to the right-hand side. Don't worry about that. So you can clearly see the difference between a positive and negative reaction. You have to do all three of these to determine the total blood type. And when you do determine blood types, you're supposed to be including, to get full credit, Rh positive or Rh negative. Is it A negative or is it O positive? And you have to have positive and negative. So there's 3 stages to that to determine the full blood type. All right. And here are our types of anemias I included in there. Different types like aplastic anemia, which they talked about that. Aplastic, iron deficiency, pernicious anemia, and sometimes I think thalassemia, they like to refer to that in the nursing program. Sometimes sickle cell. Sickle cell is, fairly common. I won't say common, but fairly common. All right, so that's what it's like to go through the notes. You can look at, I'm going to change my share to my other page. All right, hold on, let me go here. There we go. And now let me toggle back. The next time, this won't take nearly as long, folks. Okay, so here we have, now I'm back into the course page and I'm looking at that. Let me just move this thing over. The lab exercise for the week. So here we have lab number 1, and I'm going to kind of work you through some of it, and then I'm going to let you finish filling this out because it's kind of a waste of your time. I just went through all that. So here we're going to write down, determine the function of the following components of plasma. So water disperses heat. In scenario chemical, reactions can, transports and can suspend other substances, but transporting heat is a really good one. Albumin maintains an osmotic gradient. Fibrinogen, acts in clotting. Gamma globulins, are a component of an antibody, because that's what antibodies are. They have gamma globulins. All right, then we have percentage in blood. Blood or plasma is 55% of blood, normally about 55%, and the pH range of blood, you need to know for the rest of your life, 7.35 to 7.45. Normal pH range of blood, 7.35 to 7.45. I want you to know that now. All right, then we look at some slides of blood, and we also look at models. All right, so right now you can write in these functions because you can take them right from the notes. I'm going to give you a few functions very quickly right now, and then we're going to apply this to some of the images. And then you can kind of take it from there, but be able to identify the elements in the cells and functions on slides and models. All right. So an erythrocyte is a red blood cell. Function transports, oxygen and carbon dioxide. That's fine. Thrombocyte or platelet, you can say active in clotting. Neutrophils, phagocytosis of bacteria. May be first to elevate in bacterial infection. That's good to know. Most abundant, most numerous white blood cell in a normal, white blood cell differential. Eosinophils, orangey granules. I always put that in there because that's going to give you the big tip off. Produces antihistamines and combats parasites. Basophils, we generally just have you identify on the model only. Produces histamine and heparin, increases inflammation. Histamine increases blood vessel leakiness. Heparin is an anticoagulant. Lymphocytes, T lymphocytes and B lymphocytes, and then put next to B lymphocytes, produce antibodies. That's all you really have to know right now. We'll get into more of those specifics later. And then monocytes are the big mothers, right? They are phagocytize everything. Bacteria, viruses, parasites, worn out red blood cells. Right? Second to elevate with a bacterial infection. All right, so identify the agranulocytes are lymphocytes and monocytes. And the granulocytes are Ben, basophil, eosinophil, neutrophil. And here maybe just to the left-hand side of neutrophil, you put an A or a G, a granulocyte or granulocyte next to it. That might be a way. You just do it with a magic marker or something. color pencil. And what percentage, are formed elements of blood? Normally 45%. So then we go to the next page. Erythropoiesis, our feedback loop for red blood cell production. So here we have erythropoietin, that's the hormone. Right? You have red blood marrow, kidneys, increase RBC production, which increases OT levels. Those are the answers you put into, these vacant lines. So here's your negative feedback loop per se. We have a stimulus, a production site, a target site, and an action response. So I give you the stimulus. The stimulus is hypoxia / anemia. So what's the production site of the hormone in question? Red bone marrow. It targets, or actually I take that back. Oh, I beg your pardon. The production site is the kidney and it targets the red bone marrow. My bad. See, I didn't have enough Starbucks yet. And then the action response is to increase the RBC production, which increases oxygen levels. So we start out with low oxygen level capacity, right? The kidneys produce erythropoietin that targets the red bone marrow, and the red bone marrow increases the RBC production. Okay, then we move on to questions about blood viscosity. So the viscosity refers to the thickness of blood, and as viscosity increases, right, blood flow decreases. As viscosity and thickness increases, the flow decreases. So it says describe the effect of polycythemia, or an increased number of red blood cells, would have on blood flow. So if you increase the number of red blood cells, is it going to increase blood flow or decrease blood flow? It's going to decrease it, right? Because we looked at that in one of the PowerPoints. That's one reason why I have those PowerPoints, because I often address a lot of these specific questions. All right, so if we increase the number of red blood cells at polycythemia, we decrease blood flow because the viscosity of the blood increased. Describe the effect of dehydration on blood flow. Well, it's going to decrease blood flow, right? Because we said that essentially you're trying to transport the same number of formed elements in less fluid. So decrease blood flow because the viscosity of the blood is increased. And then based on 1 and 2, identify 2 major factors that influence blood viscosity and therefore blood flow. It's either your plasma and your water, and the plasma is still 55%, but your red blood cell number is increasing or decreasing. Or you have a normal hematocrit, the numbers and everything are good, but maybe the degree of hydration, the plasma is no longer 55%. So I would say the number of red blood cells and the amount of water or hydration of blood or plasma. Those are the 2 major factors. All right, then we go determine antigens and antibodies produced for the following blood types. So this is for you to fill out as an exercise. If I'm blood type A, which antigen is on my cell surface and which antibodies do I automatically produce? So we said, if we're blood type A, it means A antigens are on the cell surface and I produce B antibodies, right? If I'm blood type B, it means I have a B antigen on my cell surface and I make A antibodies. If I'm blood type AB, I have A and B antigens on my cell surface. And which antibodies do I make? Neither A nor B. And then blood type O, which antigens do I have on my cell surface? Neither A nor B. I mean, it's not like you don't have any antigens. You just don't have no A or B antigens. What antibodies? A and B antibodies. All right. And then we have here blood type testing and anti-serum responses. Remember, the anti-sera, A tests for the presence of A antigens on the cell surface, and anti-B, I should have a capital A in there. Anti-B tests for the presence of B antigens, and anti-sera RH tests for the presence of RH antigens, or anti-D for D antigens. Write positive or negative response you would expect upon administration of the anti-sera to the known blood type samples. So we're just saying here, look, if you have A positive blood, all right, we're telling you, I'm telling you what blood type you are, you're A positive. If I put a anti-sera into that well plate, would that be would you expect a positive or negative reaction? Well, if you have a antigens and I put antibodies in there, I would expect a positive reaction because you're going to get agglutination. What would happen if I put B antibodies in there? It would be a negative reaction because there are no B antigen on the cell surface. And what would happen if I put in the anti-sera RH? Would I have a positive or negative reaction? It would be positive because we've just told you that it's an A positive blood. You must have an RH or D antigen on the cell surface. So what I would expect upon administration of the antibodies or anti-sera for RH, that would be a positive clumping reaction. And so if you go through and you identify what you would expect those reactions to be, since we're not actually doing this face to face, it helps you work through what you would expect the reactions to be. And therefore, if you were given that information on a quiz or on an exam and said, hey, upon administration, because you need to know what would happen. Upon administration of anti-sera A, you see no agglutination. Upon administration of anti-sera B, you see a positive reaction or agglutination. And with the RH anti-sera, you see a negative reaction. There's no agglutination. What's the blood type? It's B negative. So by filling this out, you will have a better understanding of what would be happening when you're actually testing blood. And therefore, some of those videos, the couple of the videos that are up there are going to be helpful for this. Okay, so that's kind of quick, but let me change my share and go to some of the images. So let me just make sure that stop my share, and then I'm going to toggle back. Hold on. I'm going to go to blood notes and go to one. Okay, so you should be, hold on, let me just change my share. Okay, so I'm going to re-share this page because I went. All right, so I am back on the course page. All right, I'm over here onto the left-hand side. I'm in lab number 1. All right, so we just went through the lab notes. There are some videos you can watch on the ABO blood types and blood type anti-sera testing. They're helpful, so you understand what's happening. Then we have these other images. Now, I may use images from my PowerPoints. I may use some of the images from here on the course page. Everything is fair game. So let's look at the first image. Just shows you a bottle of albumin. All right, so what is that referring to? It's a component of plasma. What's the function? Maintains an osmotic gradient. Most abundant plasma protein regulates osmotic pressure or maintains an osmotic gradient. Now, I will tell you that sometimes, these cards will tell you exactly what you need to know, but sometimes they don't. You need to use the exercises that we filled out. Okay, then they kind of sideways, but here we have red blood cells. We have a little model on its side, and then we have the bigger one, cells on sticks, I call them. Right? So identify the cell and give a function. It would be an erythrocyte or a red blood cell, transports oxygen and carbon dioxide. That's fine. All right, here's an actual slide. This is what we're looking at, are erythrocytes. Those little dots, those are platelets there, but the pointer is pointing to an erythrocyte. All right, then we have a white blood cell on a stick. This is a model of a basophil. Right? So it says identify the cell and give a function. It's a basophil, releases histamine, heparin. Right? I would know heparin also, not just histamine. It inflames or it increases inflammation and your allergic response. That's why the histamine, that's why you take antihistamines when you have an allergy, because you're trying to offset the basophils or what's called mast cells. All right, here, ooh, look, orange granules on this model. This one, I don't like using the little one because they're not really that orangey, but I like the big one. All right, so here we have a big model with orange granules. Oh, orange granules, EO orange. It's an eosinophil, kills parasitic worms or elevates with parasites, has an antihistamine effect, produces antihistamines. Right, and then we have a slide of one. These are a little tough, but if you kind of develop an eye for it, see those granules? They're rather pale staining. I mean, there are a few purpley ones, don't get me wrong, but this one over to the lower right-hand side, and it's a little blurry, but that would be an eosinophil. Those you can see these orange granules. Okay, then we have this multi nuclear kind of polymorphonuclear nucleus to it. Here we have sausage link kind of appearance nucleus. I generally like the big one here. Don't get fooled by the orange background. Right? Identify the cell, identify the function. That's a neutrophil. Phagocytosis of bacteria, first to elevate with a bacterial infection. So when we say EO orange, we're talking about orange granules, not that orangey background. Let me just back up a couple so I just make that clear to you. Orange granules, that's an eosinophil, and it has kind of two lobes to its nucleus like that. Orange granules, eosinophil. This model that just happens to have an orangey background, it really would have been better to have it maybe pink, really more pinky or light lavender. That is a neutrophil. Okay, we're moving right along. Here's a slide of one. Okay, here we have this big U-shaped nucleus, and at this, if we had the other models of that same series that are located on the wooden base here, you would get a much better idea of the relative size of this monocyte compared to a neutrophil or an eosinophil or a lymphocyte next to it, because this is much, much bigger. This is the big mother, right? You gave me mono, so this is the monocyte. It functions in phagocytosis. Once it leaves the bloodstream, we call it a macrophage, but it can phagocytize anything. It's the second to elevate, with a bacterial infection. And that's a big monocyte right there. And you can see that, you could put about 4 big red blood cells in that, 4 to 5. Sometimes you can see the U shape, sometimes you can't. It's another one. Okay, then we have our lymphocyte. So this is a lymphocyte. Right? You have a big nucleus. It takes up about 90% of the cell. And then you have this light bluish, light lavender cytoplasm around it. Right? Not to be confused with the basophil. Right? So it's a lymphocyte. Functions in the immune response. We have T cells and B cells. B cells produce the antibodies. Right? T cells are what's called kind of the hand-to-hand cytolytic defense mechanism. But right now, just know that they're T cells and B cells antibodies for sure. And that's what it looks like on a slide. So you see how, like a basophil, you won't see this halo of cytoplasm. It'll be, first of all, darker staining, and it'll have more of a granular appearance. Here you can actually see some indentations of the nucleus, and you can see some light lavender cytoplasm over to the side. Right? That's a lymphocyte. This is a model of a platelet. It's actually a fragment of a cell, so we call it really a fragment, not actually a cell. It's active in blood clotting or just clotting. And they're the little dots. There was another picture that was probably better than this, but if you see, it looks like you took a purple magic marker and just and put a couple dots, that would be your little platelet. All right, this model is of the kidney. So we're trying to indicate, the source of erythropoietin. So when it comes to that hormone, erythropoietin, the stimulus, it says, what is a stimulus? What's the hormone produced? Right. This is where it's produced from, the kidneys. What is the target and what is the action response? So hypoxia, anemia, erythropoietin, red bone marrow comes from the kidneys, increases RBC production. So the type of question you could be asked, and let me just prepare you. Let's say you had a picture of a kidney like this, and it said, what hormone is produced by this organ? And you would say erythropoietin. You'd be asked, what's the target of the hormone produced by this organ? You say red bone marrow. What's the action or response of the hormone produced by this organ? Increase RBC and oxygen levels, something like that. What's the stimulus for the release of the hormone from this organ? Just put low blood oxygen levels, hypoxia or anemia. We do that if you haven't taken a lab at this campus. We do this frequently. We use a lot of the models and we apply those questions and things we did within the lab exercises for the week, and we apply it to models. So the same would hold true is pointing to, let me back up. Let me go back to a couple cells. I think I can only go about one at a time. Boy. I mean, you could be given a slide in the same way. So, all right. So let's say here we have this model, right? And let's say we had a picture of this model. I said, identify that cell. You would say it's a lymphocyte. Identify the function of that cell. Acts in the immune system, becomes a T cell or B cell. Yeah, that's really it. Identify or a function. Let me back up a little bit. Let me see if there's some other way I could ask you a question. I could point to a slide. It would have to be a little bit better than that, a little more clear. Identify that cell. Identify the function of that cell. I think mostly right now, I won't say all the time, it's going to be the models. Okay, so here we are. Identify that cell. It's a monocyte. Identify the function of that cell. Phagocytosis. You could just leave it at that, but you say phagocytosis of bacteria and viruses and platelets and or parasites and broken down red blood cells. Blah blah blah. I believe most of your questions are multiple choice, true, false, and the occasional fill in the blank. All right, I'm just going to work through here and see if there are any more that we did not cover. And I'm going to let you go because it's kind of lengthy to go through I think the notes are helpful to go through so you see what is there, because virtually everything you need to fill out the lab is from the notes. And some people like going through like that notes and not having to listen to a Zoom. That's fine. But at least lets me help you. The next time, what I'll probably do is just go through filling in the lab lesson and going through the images. I might go through some of the PowerPoints if if they have some good pictures, that I like to use for quizzes. All right, so here he's showing you what blood typing is, and these are our 3 well plates. You have a well plate A labeled A, B, and C. Now, what you do in theory is you have this blood that you don't know, it's an unknown blood type, you don't know what it is. So you will put a drop or two, a drop of red blood, that sample in the well for A, a well for B, and C. So a drop of blood in 1, 2, and 3. Then in well plate A, all right, with the A next to it, you put the A antibodies and you mix it up. And then where you have B, you put the B antibodies into that drop of blood. And then where you have RH, you put the RH antibodies into that little depression with the blood, and you mix it all up, and you don't double dip your toothpicks. You separate toothpick or the different ends, right? And you look to see if there's agglutination or clumping. Clumping is just, it would be nicer to say agglutination, right? So is it a positive reaction? If it's clumped, that's a positive reaction, right? And here has B, it says clumped. That's a positive reaction. And in RH, it says not clumped. That's a negative reaction. So what would be the blood type? A, B, antigens on the cell surface, no RH, you'd be AB negative. And then it shows you a couple different well plates. That's why I wanted you to know, if I knew that this was blood type, like O negative, what would essentially you're identifying what you would see in well plate A, B, and C when you put the antibodies in there. So that way you can work forward and backward. If I'm blood type O, then I would, the reaction with A antibodies would be a negative reaction. And for the B would be a negative reaction because blood type O has neither A nor B antigens on the cell surface. And if it's O negative, then you'd also have a negative reaction with the Rh antibodies. And so you can get, so then you study what is, if A antiserum negative, B antiserum negative, and B antiserum negative, if those are my results, what was the blood type? It means it's O negative. So you need to be able to work in both directions. So you have those little well plates there that you can practice what the blood types would be. And that's it. That's it. All right. And then at the bottom here, all right, so if we go back to lab 1, all right, so under the lab guides is where we have all the lab guides. Lab 1 has the notes, any videos I want to give you, where some of these other lab images may be located, all right, and then we have the blood quiz. All right. You have, I think, 1 chance, maybe 2 chances. I don't, you know what? Let me go back and look. Hold on. Let me see. Now I don't remember because I haven't done the blood. I think it's 1 chance. Hang on. I have to check them now. Yep, you get 1 chance at your quiz. And in the PowerPoint, some of those pictures, I use those pictures. I just want to tell you. But you have 20 minutes and it's 10 questions. And you get 1 minute grace period. All right. So get your quizzes. I will be Zooming, these labs ahead of time. So you can look at them at your leisure. Right, there'll be, there's a schedule up there generally. This is a summer section, so we will be doing 2 labs a week, right? And your quiz on this will be due this weekend. So get those quizzes done, and then you're going to have, like 5 days or so, or 6 days to study for your first exam, and that'll give you wiggle room in there. But we do the labs, and that weekend, your quizzes are due. And it's not, the following week. If we were meeting face to face, we would be doing it that way, but we're not. All right, so with this week, we're doing the blood lab and cardiac lab. All right, you can do them both in the same day. The quizzes are open. They are due this Sunday night. All right, just get her done. And then by the time you're done with those quizzes, I will hopefully have Zoomed the next 2 labs. I'm hoping to just do all the labs within a couple weeks here and have them so you can at least move forward through the quizzes. But the exams will be open for a specific period of time. They're not open like all semester. They'll be open for a few days. So, and if you go to the calendar, I don't know if you can still follow me here. Let me stop the share and go back again. I'm never quite sure if it sticks with me. All right, is if you go to the calendar, all right, so up on the top, we have a calendar function, all right, and it shows you the whole month, or at least mine's by month, tells you what things are due, all right. So here on the 23rd of May, you have, it says blood quiz availability ends and cardiac ends. And then the following Sunday night, systemic circulation and the immune system, right? Then we have the following week to study all my 4 labs, and actually the exam will be open. I believe I opened it on a Monday or a Tuesday. You have all week to get it done. I will tell you that probably most of you will wait till the last day to do it. That's fine, but if your internet goes out or something, you just can't come back on me. So you have all week to sit down and get this done. All right. The first exam is not honor locked, so everybody can get into step, but the last 2 exams are. So there are 3 exams. You have quizzes on each lab, do the following, the end of the week, the Sunday night, that week. All right. You just have to sit down and grind it out. All right. When you have an exam, It's going to be open, and believe I made them open for the entire week. You can do it Tuesday or Wednesday. You can wait till Saturday or Sunday, but do it when you have good internet reception. Technically, the only way I can reset an exam is if we have system-wide, St. Pete College or a hurricane, where we have major powder outages. So you really need to get it done. If you, for some reason, you get in there and it goes out right away, let's say you're on like question 2, exit the exam. Do not look at anything else. If I can go in there, because I can see every single thing that you saw, not that I want to give all my secrets away, but I can go in and see what you did every single minute that you were logged in there. So if you happen to go in there and, in the first 3 minutes or something, it's of an exam, or 5 minutes of an exam, it booted you out and you only saw like 5 questions, then immediately go right out of it, because it'll stop it. You won't be able to go back in, and you let me know, and I can go in. I say, okay, you only saw 4 questions or 3 questions or something, fine. I can reset it. But once you start looking at half and you're paging back and forth, I can't reopen that exam. You for obvious reasons, right? Yeah, I was going to say, the exams cover or will contribute 75% of your final grade, and no exams are dropped, because you only have 3 of the quizzes. I believe I dropped 1 quiz, and there might be some extra credit at the end for doing the SSI. We'd like to give you something for completing those and participating, probably be a few points on an exam. Everything is pretty much in the syllabus. Okay, and then I'm going to stop here.